So I'd also like to begin with an acknowledgement of country. I acknowledge the Wurundjeri people of the Kulin Nation and the elders past, present and emerging. So the work that I'll be presenting is really work that's been done over many years in a very fruitful collaboration between my former institute, the Centre for Eye Research Australia, and my current institute, the Florey. So I thought it would be a great topic for today. So for those of you that don't know CERA, it's a world-leading eye research institute co-located at the Eye and Ear Hospital in East Melbourne, with new premises shown beautifully there, adjacent to the Eye and Ear. And the Florey, of course, just around the corner, the preeminent brain research institute in Australia. So to make sure that we're all oriented at the outset, this is a graphical image of a cross-section of the eye. To the left of the image is the cornea, which is the clear window at the front of the eye, and light enters the eye through the cornea. It passes through a coloured diaphragm, or a donut of tissue, if you like, the iris. And the iris surrounds the pupil, the black dot that you see there. It's not actually a structure. It's just black because the eye is a wonderful absorber of light. There's not a lot of light reflecting out of the eye. It passes through that clear, Smarty-shaped structure, which is the lens, and its purpose is to converge the light, the incoming light, on the wallpaper at the back of the eye in a very precise manner. So the wallpaper of the eye is a delicate neuronal tissue called the retina. And we'll learn a bit more about the retina as we go through tonight's talk. Let me just see what's happened there. Oh, we will see. And so the nerve cells in the retina are specialised. They detect the light and convert photons of incoming light into electrical and chemical signals, which are processed within the various layers of the retina. The projections of the retinal neurons converge at a structure at the centre of the retina, the optic nerve, and they travel through to the visual centres of the brain where our perception of vision is processed. So it's a very elegant organ and one that I've devoted a large part of my life to. So for many years, the eye has been postulated as a window to the brain, and I hope during the course of this evening's talk, you'll get a sense as to why that might be the case. This graphic shows you those connections that I was indicating between the eye and the brain, and the optic nerve being the conduit for that. But in the next video, you're going to see the development of the brain and the retina in a zebrafish. So a zebrafish is a small fish that's used as an experimental model in many studies of science. And what this video will show you is just how the retina develops as an extension of the brain. So this is work done, time-lapse photography done by a group in Germany. And you can see this structure, this amorphous structure of whitish cells forming a tube-like structure. And at the top end of that tube, there are two bulbous structures. That's the developing retina. So that's actually growing out of the developing fish brain. It's forming a cup-like structure there, and that cup goes on to become the retina. So I think that's a really elegant example of how the retina develops very early in the process of brain development, and it quite literally is an outpouching of the brain. And that has significance in that as a clinician, when I examine someone's eye, I'm actually looking at part of their central nervous system. Quite a profound thought, really. There's no other part of the central nervous system that we can directly observe. And this video really is similar to the experience as an ophthalmologist of examining a patient's eye. And I remember the first time that I held a lens and was able to see inside someone's eye. And it was very much that same sense of wonderment that you have when you go snorkeling. You pop your goggles on and you put your head below the surface, and there's this beautiful world below the surface. And it's a privilege every time you get to examine an eye because it is the most gorgeous structure. So it wasn't long after the invention of the ophthalmoscope by German ophthalmologist scientist in 1851, Hermann von Helmholtz, that the association between findings on examination of the retina and diseases of the body and brain that association was made. So I wonder if anyone in the audience might hazard a guess as to what we're seeing in the top photograph there. Obviously not a normal retina. Anyone hazard a guess? That's a great suggestion, because macular degeneration often has that yellowish appearance in the center, but this is actually a patient with malignant hypertension. So someone with a blood pressure double what the normal healthy level is, over 200 millimeters of mercury. And so only 8 years after the invention of the ophthalmoscope, this German neurologist made the association between changes in the retina and a life-threatening condition of the body. And similarly, the bottom photo shows swelling of the optic nerve head. This is what we call papillary edema, and we see that in patients with space-occupying lesions in the brain. So tumours of the brain cause increased pressure in the fluid around the brain, which then gets transmitted along the optic nerve head and causes that to swell. So again, diagnosing brain cancer on the basis of an ophthalmic manifestation shows you very tangibly that changes in the retina mirror what's going on in the brain. So if you've been to an eye care provider, you would have had some sort of imaging of your retina. It's usually done very early on after you've had your vision tested or the pressure in the eye measured. Then next in line is typically a photograph of the retina. And here you can see the typical apparatus with basically an SLR camera mounted on the back of a housing and special lenses to converge the light on the surface of the retina. And as I said, the retina is the most beautiful structure, and we can see a number of things when we look at just a standard retinal photograph. You can see that yellowish disc at the center where the blood vessels are emerging from, or converging if you're talking about the veins. And that's the optic nerve. So that carries about 1.5 million axons, or the cables, the projections from the nerve cells, and carries those to the brain, carries the signals of vision to the brain. You can also see that we can distinguish between arteries and veins. Veins tend to be wider in caliber, darker in color, because they carry deoxygenated hemoglobin, which is darker in color. When hemoglobin's got oxygen bound to it, it becomes bright red. So you can tell those two apart on the basis of their appearance, but also the artery has that light reflex that you can see there. And again, very soon after the invention of the ophthalmoscope, it was noted that in mild cases of high blood pressure, you get changes in that reflex, which indicate that there's hypertension. But you can also see individual nerve fiber bundles as they course in this C-shaped structure, following the main blood vessels towards the optic nerve. So I've tried to sort of give you an indication of where they're running with those yellow lines, but you can probably appreciate better along the top of the image. Those are individual nerve fiber bundles. And there's an area at the central area shown with that yellow circle, and that's the macula. That's the region that you mentioned in macular degeneration. That's highly specialized for detailed vision, and the lens is focused on converging light on the macula. So we have at our disposal as eye care providers a vast array of different imaging technologies, and it really has exploded over the last 20 years, and it's completely changed what we can do as eye doctors. So I'm going to walk you through a couple of those now. Perhaps the most revolutionary in terms of changing clinical practice is an imaging modality called optical coherence tomography, or OCT. That's basically a scanning laser that rapidly scans the retina and allows us to produce cross-sectional images of the retina. It's a bit like the optical equivalent of an ultrasound. So an ultrasound pings out sound waves, gets them back, and it's the sort of the time of flight delay that gives you cross-sectional information about the thickness of the tissue and so on. So this is the equivalent using light, and it's just sort of a schematic showing that the machine will take a cross-section, it'll turn it on its side, and the algorithm in the machine will actually flatten the image to make it easy for interpretation. So this is what we see. So that top image is a typical OCT image. And just to put this into perspective, the center of the retina is only 1/4 of a millimeter thick. So this is tissue 1/4 of a millimeter thick, laid out, and you can appreciate with those lines that we can see the individual neuronal layers within the retina. So this is an extremely powerful technology that allows us to interrogate almost at a cellular level what's going on in the retina. This is technology that's now available in almost every optometry practice, certainly every ophthalmology practice, and it's completely changed our understanding not only of eye diseases, but also of brain diseases. A recent advance on that technology is something called OCT angiography. So you might have heard about angiography. You might even have had an angiogram, which is typically where we inject a dye, usually a fluorescent dye, intravenously. And in the case of an eye angiogram, we would take a series of photographs with a blue light. The blue light would excite the fluorescent dye. We'd get green fluorescence from that dye, which we'd capture using a certain filter, and we'd be able to see the delicate blood vessels in the eye. And that's important because many eye diseases are associated with changes to blood vessels. But OCT angiography means that we can now do that without needing any dye at all, which is very convenient and much safer for the patient. And that's based on motion artifact, motion contrast with little red blood cells zipping through the blood vessels. So it only shows us vessels that are perfused. But that's exquisitely detailed. If we hone in and blow that area up, this is the fine capillaries in the central area of vision of the macula. So we can see down to even the smallest blood vessels. The relevance of this to the brain is that there are very, very close analogies between the blood vessels of the retina and the blood vessels of the brain in terms of their structure, their barrier properties. And so we can learn a lot about the brain by looking at blood vessels in the retina. So these imaging advances and others have led to the emergence of an entirely new field called oculomics, which is where associations are made between findings from eye imaging and systemic diseases. Most of the time, these are association studies. You take 1,000 people with a particular disease, you image them on a range of different imaging modalities, you identify what features are associated with that condition. And there are a number of relatively robust associations that work at a population level. At the moment, they're not particularly predictive at the individual level, so it is a little bit of a failing. We are, however, seeing dramatic advances in the power of artificial intelligence in picking up very subtle patterns that we as human observers are not very good at gleaning to develop biomarkers that are specific to a particular condition. So this is a rapidly emerging field. We recently published a review on this. But you can see the range of conditions that you can get information about by imaging the retina, about heart disease, stroke risk, anemia, high blood pressure, high glucose, changes in your lipids, even liver diseases, kidney disease, and many more. I'm particularly interested in diseases of the central nervous system. So the rest of the talk is going to focus on work that we've done looking at Alzheimer's disease. So you've probably heard of Alzheimer's disease. It's the leading cause of dementia globally. And it's a devastating condition in which there's progressive loss of initially learning capability, then memory, changes to personality, and it is the leading cause of death in most developed countries now for women, and second leading cause of death for men. Affects around 50 million people globally, and the number of people affected is expected to increase significantly in the next few years with aging of the global population. It's a huge socioeconomic crisis. Health systems are already at bursting point, and Alzheimer's disease risks tipping that over in the next few years. Now, we understand a lot about Alzheimer's disease, but there are still significant gaps in our understanding of how these things all piece together to actually cause dementia. But what is beyond contention is the fact that there are hallmark changes, one of which is the accumulation of a sticky protein called amyloid beta that accumulates between the nerve cells in the brain. That accumulates. It's very hard for that sticky protein aggregate to be cleared by the immune system. We know that amyloid can lead to impaired connection between neurons, and so the information exchange between the nerve cells in the brain gets impaired. We also see the abnormalities in one of the scaffolding proteins within nerve cells called tau. That becomes also sticky and aggregates and forms tangles within neurons and ultimately can lead to their death. So those are the sort of hallmark changes, but we also see a range of other changes in the brain in Alzheimer's disease: activation of immune cells, disruption of the blood-brain barrier, accumulation of iron. And so these are all hallmark features. What the end result of that, though, you can see in the cartoon, you get death of nerve cells in the brain and shrinkage of the brain. So the important thing to realize with Alzheimer's disease is that the changes that accumulate in the brain do so very slowly over a prolonged period of time. So these graphs are from studies of the very few people that have genetically defined Alzheimer's disease. Most Alzheimer's disease is what we call sporadic. There's no single gene that causes the disease. So 95% or more of people with Alzheimer's disease may have some genetic risk factors, but it's not if you have that gene, you will get Alzheimer's disease. But 5% are genetically determined to go on to get Alzheimer's disease. So we can identify these families, which means that we can study people from very early age and we can track the changes that occur in the brain. And so with the generosity of these families that have participated in this research, we've understood that it's about 30 years from the onset of the first changes in the brain to the beginning of memory impairment, which is the dashed line at 0. So about 30 years. And you can see the purple graph shows the accumulation of that amyloid protein in the brain, that sticky protein. So what that means is that there's this big window of opportunity to detect people who are on a trajectory to get dementia. And if you are to intervene and make a meaningful difference in this disease, you've got to get in before symptoms start or are too advanced. So there's this big opportunity there. And I should add that a lot of the work to understand that trajectory was done here in Melbourne with Chris Rowe and Colin Masters. So Melbourne has been really an epicenter of understanding of Alzheimer's disease. So the way in which people traditionally are diagnosed with Alzheimer's disease, it's typically a presumptive diagnosis based on a series of tests of memory function and brain function by a specialist, usually a neurologist or a geriatrician, a psychiatrist. And that has about 70% sensitivity in the best hands. So about 30% of the time they're wrong. And that's because presentations can be quite varied. If you're lucky enough to be in a study of a new drug, you might be invited to have a specialized brain scan called a PET scan, where a specific antibody with a radioactive tracer is injected intravenously. That antibody binds specifically to amyloid, and then you go in a scanner and you can work out actually the amount of amyloid in the brain. There are now scans for tau as well, but not many people get access to that because it's costly. There are not many scanners with the capability. The other way that you could get a diagnosis is by having a spinal tap or a lumbar puncture, which is where a needle is inserted in the back to collect some of the cerebrospinal fluid that bathes the brain and the spinal cord. And that's then sent off to the laboratory for specialized tests, looking again for amyloid and tau. You can imagine that's not a procedure that many people willingly subject themselves to. It has to be done in a hospital and so on. So again, it's not really done at scale. So there has been this big demand for scalable, non-invasive biomarkers of Alzheimer's disease. When we started this work about 7 or 8 years ago, we hypothesized that perhaps this link between eye and brain might be something that we can exploit for Alzheimer's disease diagnostics. The field has moved along rapidly in the last few years, and now blood biomarkers of Alzheimer's disease are coming to the fore. And it won't be long until you can get a simple blood test that is actually very, very sensitive and specific for Alzheimer's disease. So that there'll be a transformation in the next couple of years around determining where you are on that trajectory to Alzheimer's disease. You might say, well, why on earth would I want to know? Well, we are entering a new age where there are actually disease-modifying therapies for Alzheimer's disease. The first antibodies to target amyloid beta in the brain have been approved in the US. They slow cognitive decline by about 30% if patients are started in the early stages of Alzheimer's disease. So we're entering a new age of hope for Alzheimer's disease. But anyway, we were very interested in whether the eye could serve as a window to the brain. for Alzheimer's disease in particular. And this is work that was published around the time we became interested in this. And this is brain tissue from 2 patients who've died with Alzheimer's disease. And it's examined under a microscope after it's been stained with an antibody that's attached to a particular chemical that gives a rusty stain if the antibody's bound. So it's a way of showing if amyloid is in the tissue. And you can see in both of those cases, there are these clumps of amyloid in the tissue, which helps to confirm that they have Alzheimer's disease. So this group was interested to see if similar changes occurred in the retina. And indeed, they did show that the same rusty staining for amyloid is present in the retinas of people with confirmed Alzheimer's disease, shown in the yellow box, but not present in the retinas of people who were age-matched but died without Alzheimer's disease. So this suggests that indeed amyloid beta is accumulating in the retina as it does in the brain. There are some differences. The size of the accumulation's a little bit smaller in the retina. But this speaks to the fact that the retina can be a window to the brain for Alzheimer's disease. The problem is that the accumulation is small in quantity, and without that dye, there's no way you can see it on conventional microscopy. So there's no chance of detecting it with conventional imaging. So our team was very interested in whether a new experimental type of imaging called hyperspectral imaging might allow us to see amyloid in the retinal tissue. So to explain hyperspectral imaging, I'm going to take you back to your high school physics, and you might recall this image, which Isaac Newton, had that classical experiment where he had a dark room, made a little hole in the curtain, and so there was a beam of sunlight which he passed through a prism and showed that he could split white light into its component parts, the colours of the rainbow, which is quite a mad thought, isn't it? That you can take white light and it's actually a combination of all the colours of the rainbow. And what that experiment showed Newton, or confirmed for Newton, is that light is composed of electromagnetic radiation of a certain group of wavelengths. So violet and blue light, what we call short wavelength light, has a narrower distance between the little peaks of the wave, whereas longer wavelength light, like red light, has much longer periods between those peaks and troughs. So, and that's a physical property of those wavelengths of light, and our perception of colour is a human construct. That's not intrinsic in that light. We see that spacing in those wavelengths of electromagnetic radiation as colours because of the chemistry of the cone photoreceptors in our retina. So colour is our interpretation of the wavelength of light. So it's so innate and so fundamental to our experience that it's hard to entertain the idea that colour is just a perception and it's not a physical a physical feature of that light. It's our interpretation of this difference in wavelength. So you might say, well, that's fascinating, but how can you use that for the purposes of imaging? And so the way that we do that is, instead of using conventional photography, where we just have a white light flash, we collect all the light that's bouncing off the retina, what we do in hyperspectral imaging is that we sequentially image the retina with different colours, or different wavelengths, if you like, of light. So we go all the way from violet all the way through to infrared. And at each time, we capture an image on the sensor of light bouncing back at that different wavelength of light. And we are interested in that because different wavelengths of light have different physical interactions with whatever we are that we're imaging. So we can get much richer information of the composition of what we're imaging when we look sequentially across different wavelengths. So what we end up getting is a stack of images across different wavelengths or colours of light, and we measure the reflectance at each point. So I could go to any pixel in that image and get a readout of reflectance intensity as a function of wavelength. So we get a fingerprint for that location of the retina, which speaks to the details of its structure. So put another way, by using different wavelengths of light to image the eye, we get to see things that have never been possible to see before. So here's a video, I should start, of a patient getting hyperspectral imaging. So it's a second long acquisition there. It's just repeated on loop there. I'll just do it again. So in that second, there's actually 91 different images being acquired at different wavelengths of light across the visible spectrum. All you'll probably see is red, green, and blue, and that's speaking to the biases of our cone photoreceptors, which have peak sensitivity for those wavelengths. So, but I can assure you that if we slow it down and step through, you'll see 91 different colors of light. So that's how it is for the patient, and most of the patients describe it as a quite a beautiful thing. You sort of see the almost kaleidoscopic colors. But the notion of imaging across different parts of the electromagnetic spectrum is nothing new. Just reminding you that visible light occupies a very small part in the middle of the electromagnetic spectrum, which goes all the way from gamma rays through to radio waves. And imaging across different wavelengths of light, if you like, is the basis of modern astronomy. We have space telescopes that are sensitive to different parts of that spectrum, because when we combine the information that they acquire across that range of wavelengths, we get much richer information about distant galaxies and stars. So here's a tangible example of the nature of the information that we get when we image across different wavelengths of light. This is an image of the pillars of creation, a star-forming region in the Eagle Nebula, which is about 6,500 light years from Earth. And with the Hubble telescope, which is sensitive to visible light and ultraviolet light, we see clouds of gas and dust. We see a few stars in the stellar background. If we then look with the James Webb telescope, which is sensitive to infrared light, we see many more stars in the distant background. Many hundreds of thousands of stars that we can't see with the Hubble, and that is in part due to the fact that we're looking across different wavelengths of light. So back to the question of imaging the retina with different wavelengths of light. We know that amyloid is in the retina, so we wondered whether we could use hyperspectral imaging to get a signal for amyloid in the retina. At the time that we were doing this, a group in the US had a similar idea, and they started off by looking at Alzheimer's disease model mice. So we can take mice and genetically engineer them to express the mutations that are responsible for genetic Alzheimer's disease, and they overexpress amyloid in the brain, and it turns out that they also express it in the retina. So we can take the retinas from these animals and look at it under a microscope with a hyperspectral camera, and at 2 months of age, the Alzheimer's mice in red and the normal non-genetically engineered mice, the so-called wild-type mice in green, overlap. The reflectance from the retina is the same for both animals, but over time, there's a difference that emerges as those mice start to get accumulation of amyloid in the brain and the retina. And you can see at 6 months, there's a big separation in those curves, and at 8 months, even greater. And what we see is that there's less light being reflected from that retinal tissue in the shorter wavelength range, the sort of 450 to 500 nanometers in the blue to sort of greenish yellow. So they then went on and had a look at tissue from the brain and retinas of people who died from Alzheimer's disease, again plotted in red, and people who died from other causes, age-matched. And they found that same separation in the reflectance of light from the brain tissue and the retinal tissue. So there's less short wavelength light being reflected from the tissue of individuals with Alzheimer's disease. And what they hypothesized is the accumulation of amyloid protein, that sticky protein, between neurons in the brain and in the retina scatters light in a wavelength-dependent manner. So there's less short wavelength light, blue light, being reflected back to the camera sensor. More of it's being scattered within the tissue. So our team had the fortune of securing some generous philanthropic support to buy the only camera in the world with the capabilities of doing hyperspectral imaging of the retina. The retina is actually quite hard to image, optically. And so we had the fortune of getting a prototype camera, and we asked the question as to whether we could do a similar thing to what the US group did on post-mortem tissue, but in living individuals. And so to do that, we recruited patients from a very famous study of aging called the ABLE study, which is based in Perth and primarily in Melbourne. And individuals in the ABLE study undergo a battery of tests, memory tests, cognitive tests, and then they have the option of undergoing a brain scan, the PET scan that I mentioned, that can image the amount of amyloid. You can see the graphic there where red equals amyloid. And so we recruited people who had early Alzheimer's disease and people who'd had the same battery of tests but were cognitively normal. So no Alzheimer's disease and no amyloid in the brain. And we asked a simple question: can we take a hyperspectral retinal photograph and can we separate out those two groups without knowing which group they belong to ahead of time? This is a significant challenge. So remember in the OCT scans, I showed you that the retina is a multi-layered structure. It's a bit like imaging a lasagna, right? Imagine if I said, we're going to image a lasagna from the top down and we want to pick up all the oregano in that lasagna. You can imagine that there's quite a signal processing challenge here because there's so many other components in that retinal structure that will have an influence on the reflectance of light. So that was the nature of the challenge. And that man in the corner there is Dr. Xavier Hadou, who's a senior postdoctoral scientist and engineer who I recruited to my team, who had world-leading expertise in signal processing for hyperspectral data. His PhD was flying drones over crops in France equipped with hyperspectral cameras to work out what proportion of the crop is weed versus crop, what the nutritional requirements are on the basis of video hyperspectral signals. So he is a world expert in that. So going back to our patient group, we had 15 individuals who had early Alzheimer's disease, who'd had the PET scan, were positive for amyloid in the brain, 20 individuals who were cognitively normal, no amyloid in the brain, and they were comparable for all potential confounding factors, eye disease and so on. The only way that they differed was in their performance on the memory test. As you can expect, the people with early Alzheimer's disease had lower scores. Now, we imaged them all on our hyperspectral camera and we plotted the data. This is raw data, and red being people with Alzheimer's disease, early Alzheimer's, and blue being our controls, our cognitively normal people. And you can see that there's a trend for a difference between those groups, but there's a lot of noise within each group. And now we weren't surprised by that, and I'll show you why that was. These are two perfectly healthy individuals, and you can notice a profound difference in the appearance of their retina. The person on the left has very fair skin. The person on the right has darkly pigmented skin. So the amount of melanin underneath the retina has a profound influence on the light reflecting back when you take a photograph. So we knew that there'd be a lot of within-group variability that we need to control for to detect the very subtle signature for amyloid in the retina, a bit like, controlling for varying amounts of bechamel sauce in the lasagna to work out the oregano. And so Xavier applied a very conservative mathematical technique called dimension reduction by orthogonal projection, which is a way of reducing that within group variability while maintaining the between group variability. So we don't want to separate out, lose the faint signal for amyloid in that process. And what you can see there is that those curves are significantly different. They're separated, and particularly in that shorter wavelength range. And remember that the studies from the mouse and from the post-mortem data also showed that most of the difference was happening in that short wavelength range. It's hard to analyze data from complex curves like that. So Xavier reduced those down to a single score using a mathematical method, which he published for each participant. And what you can see there is that when you plot those scores for people with Alzheimer's disease versus people without Alzheimer's disease, so red versus blue, they separate out. And if you look between the eyes, you see similar findings. We also had a validation group, which is the group across to the right, which is individuals who'd had a PET scan, again, cognitively normal Alzheimer's disease. They were imaged by our collaborators in Canada on the same type of camera. They sent us their data without us knowing which group participants belonged in. We applied our mathematical correction and we were able to separate out those individuals. So that suggests that a retinal image can be used to identify or discriminate between someone who is cognitively normal and someone who has early Alzheimer's disease. In order to make sure that this was in fact correct. We wanted to go back to the laboratory and make sure we could see this in mouse models of Alzheimer's disease. And for this work, we collaborated with fantastic scientists in the Department of Optometry at the University of Melbourne, and they had Alzheimer's disease model mice. We had a hyperspectral camera that was modified to image a live mouse eye, which is, again, no mean feat. And so we imaged aged mice, control mice, healthy mice, and Alzheimer's model mice, and we applied the human algorithms, the human mathematical correction, and again, we could reliably separate out those groups. Unlike humans, we could take the mouse eyes and then process them for examinations under the microscope, again, staining with that rusty stain, the antibody that's specific for amyloid. And in all of the Alzheimer's mice, we found amyloid in the retina, and in none of the control mice, we found amyloid. So again, that was another point of validation. So that's all good and well. You could say, Peter, I don't need a fancy retinal scan to tell me that someone's got cognitive impairment. Can you actually use this to plot someone along that trajectory of disease? That's the ultimate question. Does the amount of amyloid or the signal that you're getting for amyloid in the retina predict what the load is in the brain? And so to interrogate that question, we recruited a further 112 participants from the ABLE study, all of whom had undergone PET scans. And we wanted people across the severity spectrum. We had 20 people with Alzheimer's dementia, advanced dementia. We had 30 individuals with the early stages, what we call mild cognitive impairment. We had 41 individuals who were cognitively normal, so normal brain function, and negative for amyloid on the brain scan. But importantly, we had 21 individuals who were cognitively normal, but positive for amyloid on the brain scan. About a third of those who were positive for amyloid in the brain scan do not yet have Alzheimer's disease. They're at higher risk of going on to get Alzheimer's disease, but we thought it was really important to include that group so that we could work out if our signal was specific for amyloid accumulation and not detecting some other surrogate marker like loss of nerve tissue in the retina. I'll speak of the details of this, but we used a deep learning model to detect patterns in the data. Deep learning is a type of artificial intelligence that's very good at gleaning complex patterns in data sets, and Xavier has expertise in doing so. So we developed a deep learning model, and we set it with the task of saying, taking retinal image data alone, can you predict the quantitative score for the amount of amyloid in the brain? There's a way of taking those PET scans and computing exactly the amount of amyloid in the brain. So could we predict the amount in the brain based on just a hyperspectral retinal photograph? And these are the results. So we're plotting our estimated brain score on the basis of the retinal scan on the vertical axis and the actual measured amount of amyloid in the brain on the horizontal axis. Now, if there was perfect concordance between our estimate from the retina and the actual measured amount in the brain, all of those points would align perfectly with the diagonal. You can see that we're not too far off. So that suggests in most cases, in this relatively small data set, the retinal signal correlates with the amount of amyloid in the brain. So this is a new imaging technology. We're one of the first groups in the world to have a go at looking with hyperspectral imaging. And as an ophthalmologist, I'm very interested not only in brain diseases, but in eye diseases. So we have imaged, we've now got over 30,000 hyperspectral retinal images in our database across a range of blinding eye conditions, glaucoma, diabetic eye disease, age-related macular degeneration and inherited eye diseases, as well as eye tumours. And we've detected biomarkers that we can't see with any other form of imaging with hyperspectral. For instance, with the tumours at the back of the eye, I can show you a slide later on. One of the challenges that an optometrist has is that 7% of the Caucasian population has freckles at the back of the eye, like freckles on the skin. And just like freckles on the skin, a proportion of those can go on to become melanoma. And melanoma in the eye has an abysmal prognosis, 50% mortality in 5 years. It's one of the poorest prognosis tumours. So you can imagine that if 7 to 10% of the Caucasian population have these freckles, they need to be monitored annually. It's a huge burden separating those out. And one of the challenges is that we're not very good at delineating the boundaries, because sometimes these freckles are variegated. They're a bit brown, but they also have lighter regions. And we know that detecting the boundaries of those tumors is very difficult. So we've shown that we can very accurately with hyperspectral imaging detect those far more accurately than even a qualified ophthalmologist, and as well as a ocular oncologist, so a specialist in eye tumors, using every available imaging modality they have. We can do that automatically in a fraction of a second. So in the last couple of minutes, I thought I'd just tell you about the work that we've done around developing our own hyperspectral camera. So the camera that we had for these studies was extremely capable, but the analogy that I'd have is a bit like having a Ferrari to go and get your groceries. You can do it just as well as in a Toyota Corolla. You don't need the Ferrari. So that was a very expensive piece of kit. Quite fragile, not a clinically robust instrument, needs an expert operator, and perhaps most importantly for the patients, requires a very large pupil diameter. So you need at least a 6 to 7 millimeter pupil, which for most older patients requires several rounds of eye dilating drops. So we have to pretty much marinate our patients in drops for 45 minutes before their pupils are big enough. And then they stay big for a long time. And most people who've had their pupils dilated don't enjoy the experience very much because you're very sensitive to glare. So these were important parameters around designing our own lower cost, more clinically appropriate camera. We wanted to do one that didn't require pupil dilation at all. I think we were a bit naive in hindsight. We started thinking that this might be quite a simple thing. It turned out to be a long and challenging journey. We started with a typical sort of physics lab setup with lenses on a breadboard. We showed proof of concept using a 3D printed prototype shown there. Again, the tolerances with printed plastic is just nowhere near tight enough to get the precise shaping of the light that you need to effectively do this. But we showed enough proof of principle that we could put in an application to a philanthropic granting body. We secured funding, which allowed us to develop a far more sophisticated machine. I know that this looks like a PC tower, but within that is a very sophisticated machine that has allowed us to image the retina across 30 wavelengths of light in a quarter of a second. There's a hard sort of physiological constraint because you've got the latency of the pupillary reflex is around about 0.3 of a second. So if you're going to flash a bright light into the eye, your pupil's going to constrict and then it's game over. So we needed to acquire hyperspectral images within the latency of the pupillary reflex. And so we set that at 30 images, and we actually showed that we can get similar sorts of information as we can with 91 images. Here's a video rendering of the device that we've ended up building with a medical device development company helping us called Ingenuity Designs. A semi-autonomous machine that an operator can just press on the screen. It finds the eye, it finds the optimal focus, it acquires a hyperspectral image, and we're in the process of commercializing that. We can produce these devices for the same sort of price as a standard retinal camera, and yet we can get vastly richer information about the retina. This device was developed really in close consultation with many optometrists and orthoptists. It's had a lot of interaction with clinicians and scientists. So with that, I'd like to acknowledge the tremendous support we've had from numerous funding agencies over many years. It all really started with support from the Ugelbar Foundation, with thanks to Bales Meyer and Samantha Baillieu. But we've had support from the Australian government through the NHMRC, the EU, the Alzheimer's Drug Discovery Foundation, the National Foundation for Medical Research and Innovation, and the Medical Research Future Fund through the BioMedTech Horizons grant. It's been a long journey with many invaluable collaborators, numerous very clever scientists at the Centre for Eye Research Australia, the University of Melbourne, and external universities. We have cameras now in Belgium and Sweden and New Zealand and several in Australia, and we're actively gathering data. And the latest thing that we're doing is a special type of artificial intelligence called building what we call a foundation model, which is an artificial intelligence network that is very good at recognizing the spectral characteristics or the light reflections characteristics in normal, so that you only need relatively small data sets of disease for it to recognize that far more efficiently than building a separate model for every disease. And so we really see that is the future of this technology.